Good day, everyone. My name is Almara J. Carbon, and together with my other group members, Dorothy Ablan, Serlene Cabanit, Almira Fegabales, Zeneth J. Ordonio, and Hadassah May Paloma, will be discussing the quality assurance in parasitology laboratory. So here are the lesson intended learning outcomes. First is to understand the quality assurance program for parasitology, to identify the three stages of quality assurance in a parasitology laboratory, and to know the appropriate parasitologic techniques as well as diagnosis of parasitic infections. Quality assurance is a method that aims to improve the reliability, efficiency, and usage of laboratory services over time. Procedure manuals, quality control for tests, reagents, and equipment, workload, work environment conditions, training, and laboratory staff assistance are all elements that affect laboratory performance. It is an important aspect of clinical laboratory practice that can be achieved by first, internal quality control, which involves laboratory employees evaluating their own work using a set of procedures. Internal quality control allows the laboratory to examine its own procedures and verifies that the test was completed to the best of the staff's ability and with the utmost care. It can be done as often as needed and is less expensive than external quality control. Next is the external quality assessment, in which an outside party or agency assesses the laboratory's performance objectively and on a regular basis. External quality control detects systematic flaws in laboratory procedures early, highlights areas of development, identifies training requirements, offers objective verification of testing quality, and permits comparison of performance and findings across test sites. Next is Mr. Zeneth Ordonio, which will be discussing the quality assurance program for parasitology and the three stages of quality assurance in a parasitology laboratory.
Quality Assurance Program for Parasitology. A number of parasitology quality assurance systems have been established in various nations, with the United Kingdom, National External Quality Assurance Scheme, or UK NICAS, for parasitology serving as an excellent example. By flagging problem areas and offering targeted teaching and training, it has enhanced parasite infection awareness in UK laboratory practice. The UK NICAS was created with the goal of improving parasitic disease diagnosis by examining samples from patients with parasitic infections, providing training material depicting odd or uncommon parasites, and focusing on areas where low performance was observed. The UK NICAS now has the following sub-schemes: fecal parasitology, including extraintestinal parasites; blood parasitology, including tissue parasites; toxoplasma serology, and an extractional sub-scheme. A Quality Assurance Program, or QAP, is a criterion for obtaining a license in the Philippines. Internal and external quality assessment programs are required as part of the QAP. Internal quality control methods should be implemented in each laboratory according to the internal quality assurance plan. Quality should be pursued by all laboratory personnel, regardless of their hierarchical standing. It is necessary to determine the current state of the laboratory, identify any deficiencies, and carry out the necessary procedures. A clinical laboratory is required to participate in the National External Quality Assessment Scheme, or NICAS, run by designated national reference laboratories under the external QAP. The NICAS, or National External Quality Assessment Scheme, is a schematic quality assessment of laboratory processes conducted by an external agency using materials with known but hidden outcomes. It is carried out to ensure that laboratory processes are followed to the letter of the law and that laboratory results are correct and within the acceptable range for providing high-quality healthcare. The Research Institute of Tropical Medicine or RITM is a reference laboratory for parasitology, laboratories conducting NICAS or National External Quality Assessment Scheme. 3 stages of quality assurance in a parasitology laboratory. In a clinical laboratory, quality assurance or QA refers to the entire testing process, from the ordering of a test to the interpretation of the results. Quality assurance encompasses all processes required to provide reliable results. It is broken down into 3 stages: pre-analytical, analytical, and post-analytical. The pre-analytical stage involves activities that influence the quality of laboratory results before the actual laboratory operation. These operations include training of test employees, patient preparation prior to specimen collection, specimen quality and volume, and specimen processing and labeling. The analytical stage or step involves technical or laboratory processes done to achieve correct test findings. It encompasses normal work organization, the types of tests and reagents, the state of the equipment, and standard operating procedures. It also covers all areas of quality control, such as remedial actions to be taken when mistakes in the findings are discovered. And lastly, the post-analytical stage or step comprises the correct and accurate reporting of results. It covers the structure of recording, reporting, and interpreting findings, as well as the speed with which they are reported.
Good day everyone, this is Almira Fabi Gabales, a member of Group 3, and I am tasked to continue the discussion and elaborate the topic personnel, specifically those who are responsible for the quality assurance in a parasitology laboratory. Now, let's start. The laboratory supervisor has overall responsibility for quality assurance in a diagnostic parasitology laboratory. The qualifications of the supervisor must be consistent with the existing policies on the operation of a clinical laboratory. The supervisor should ensure that, first, a procedure manual is available. Second, records are properly kept. Third, controls are available for diagnostic procedures. Fourth, equipment and instruments such as microscopes, incubators, centrifuges, etc. are properly functioning and calibrated. Fifth, clerical and analytical errors, if committed, should be corrected. Sixth, unusual laboratory results such as uncommon parasites, unusual antibody titers, etc. are checked. And lastly, standardized procedures are being followed in the laboratory. Additionally, all laboratory personnel should be trained on the different aspects of running a parasitology laboratory. The person in charge of providing instructions to the patient must be familiar with all aspects of specimen collection, including preparation of the patient, specimen collection times, sample quality and volume, condition of specimen container, use of preservatives, and proper labeling. Lastly, laboratory staff must be familiar with the appropriate diagnostic procedures to be used for each type of specimen and parasite, and must be competent in morphologic recognition and differentiation of parasites. And now, in this portion of the video, I am going to discuss the topic Procedure Manual. A procedure manual must be made available to the laboratory personnel for reference. This should contain the following information. First, instructions for proper collection and handling of samples. Second, information on when to reject parasitologic sample, such as inadequate amount of specimen. specimen not labeled with full name or ID number, improperly preserved specimen, and etc. Third, preparation of reagents and solutions. Fourth, detailed descriptions of techniques. Fifth, criteria for identification of parasites. Sixth, quality control procedures. Seventh, reporting and interpretation of results. Eighth, general safety precautions such as use of gloves, laboratory gown, proper disposal of specimens, and proper handling of inflammable and hazardous reagents, etc. For additional information, in a study that assessed the quality assurance of a number of clinical laboratories in Iloilo, Philippines, 65.5% of laboratories visited were shown not to have any manual of procedures available as reference, which should not be the case.
Good day, everyone. This is Hadassah Macy Paloma, and I apologize for the way I speak. I'm currently not feeling well, but I hope you can still hear my words clearly. So let us now proceed to the instruments and equipment. Every diagnostic parasitology laboratory needs to be equipped enough to guarantee efficiency of their tests. Preventive maintenance of instruments and equipment should also be done routinely to ensure that all instruments and equipment are functioning properly and are in good condition. So, one of the most important instruments in diagnostic parasitology laboratory is the microscope. So the microscope needs continuous care to maintain its good functioning condition. Its condenser alignment must be regularly checked, and it needs to be protected from dust, vibration, and moisture. Take note that heat and humidity can contribute to fungal growth, which damages the lenses. So it is important or necessary that it must be clean. The lenses must be cleaned from time to time with the use of lens tissue, not just any ordinary tissue, for it may cause scratches in the lenses. Next is microscopes should also be calibrated with the use of an ocular and a stage micrometer in consideration of identifying protozoan cysts in its particular sizes. Another important equipment in the diagnostic parasitology laboratory is the stereoscopic microscope, which offers easier examination of large specimens like adult worms and worm segments, including arthropods. Next, we have the centrifuges, which are also needed to be calibrated for appropriate speed of use. A properly calibrated balancing tube at opposite buckets can prevent damage to the centrifuge and breakage of the test tubes. The inner walls of the centrifuge must also be wiped with antiseptic every after use. Next is a standard thermometer, which is also needed in order to check the temperatures of refrigerators, incubators, water baths, and freezers. Next, we have the fume hood, which is also necessary in a diagnostic parasitology laboratory, for it is where volatile and toxic chemicals like ether and formalin are done for diagnostic procedures. Some additional items included, which are necessary in a parasitology laboratory as well, are the pH meter, the differential counter, and glasswares. Glasswares includes the microscopic slides, volumetric flasks, beakers, funnels, drop bottles, pipettes, and test tubes. Last is we have the disposables. The use of disposables, gloves, syringes, and needles are also recommended as the problem of parenterally transmitted organisms are increasing. So the next topic would be reagents. Take note that when in practice, not all reagents need periodic review, but antigen, stains, and fixatives must be checked before using. First, we have zinc sulfate solution, which are reagents for concentration techniques, which may be needed to be checked for specific gravity. Next is the buffer agents, must also be checked for its pH level. There are practices and rules that we need to follow in consideration of our reagents. So all reagents should be labeled properly and must have preparation and expiration dates. We must also keep in mind that there are some reagents that must be kept in sealed containers or dark bottles. This is to prevent degradation. Next is observe special precautions when handling and storing explosive chemicals such as phenol crystals and flammable solvents such as xylene, ether, and acetone. Lot and or batch number of reagents needs to be properly noted for commercially purchased reagents. Avoid overstocking of commercially prepared staining used for blood parasites. Stain quality must be checked using positive slide for control at least once with every new batch of stain. Lastly, the usage of control reagents are highly recommended, especially sero-immunodiagnosis.
Good day, this is Erlene Priscilias Cabanit, and I'm about to discuss the appropriate parasitologic techniques and reporting. So let's start with the appropriate parasitologic techniques. So the employment of proper parasitologic techniques in the lab will aid in the accuracy of diagnostic or process outcomes. The laboratory technique used may be determined by what is being considered as part of the differential diagnosis and the specific objective of the laboratory investigation. So for instance, diaric stools that might contain Entamoeba histolytica or the Giardia duodenalis may be best examined using direct fecal smear. So a number of intestinal helminths are better demonstrated using modified cathotic smear method than by direct fecal smear. So routine parasitologic screening and diagnosis 356 medical parasitology in the Philippines in health centers and hospitals may make use of combined direct fecal smear and the modified cathotic smear method to increase the chances of catching true intestinal parasitic infections. So food handlers are best screened using formalin ether ethyl acetate concentration technique or the FEC than by direct fecal smear or the modified cathotic smear method. So modified quinine stain may be better demonstrate intestinal coccidian infections, and a clinical trial using new helminthics against soil-transmitted helminth or the schistosome infections benefit most from the use of CatoCats method that allows quantitative diagnosis in terms of egg counts. So control programs for soil-transmitted helminth and the schistosome infections may utilize CatoCats method to demonstrate baseline and follow-up parasitologic parameters. So all the health center laboratories in Iloilo, Philippines utilize direct fecal smear or the DFS technique for the parasitological diagnosis, while only 3.7% of the health centers laboratories may use of the cathotic smear method, which is the CathoCats method or the FECT. So what you can see is the recommended stool examination techniques for this specific situation. So the specific situation is the routine clinic or the hospital stool examination. And its recommended stool examination technique is the direct fecal smear and the modified cathodic method. Next specific situation is the examination of the diarrheic stools. So the recommendation examination technique is the direct fecal smear. Next is the screening of food handlers. Its recommended stool examination technique is the formalin ether ethyl acetate concentration technique. So in terms of the screening of the overseas Filipino workers, the recommended stool examination technique that's going to be used is the formalin ether ethyl acetate concentration technique. So for the solely transmitted helminthiasis or the schistosomiasis surveillance, it's the CATO-CATS technique, while the epidemiologic investigations uses direct fecal smear, modified cathodic method, CATO-CATS technique, and the formalin ether ethyl acetate concentration technique, depending on the parasitic infections being investigated. So let's move forward to reporting. So the results of a laboratory examination must be treated as private documents shared only between the laboratory and the requesting physician. So all laboratory requests as well as the findings must be carefully recorded in a logbook or a spreadsheet that only authorized workers access. So the data of examination and reporting of results should also be properly recorded. So qualifying statements regarding the quality of specimens such as inadequately preserved when received in the laboratory or contaminated with water and urine should also be noted. So in the reporting of results, the complete name and of the parasite must be mentioned and the specific stages. So for example, it's ova, larvae, adults, cysts, trophozoites, and etc. must be indicated. So the presence of the charcoal-laden crystals or the CLC or budding yeast cells must be noted too. So the CLCs indicate an eosinophil response and are usually associated with allergic or parasitic disease. There is no common convention for the reporting units of the CLC. It is reasonable to report them on presence or the absence of the basis only. So likewise, the presence of the macrophages, the eosinophils, and the polymorphonuclears is important information for a clinician. So quantitative reporting of the fecal leukocytes per high power field is recommended. So that is WBC over HPF. However, caution must be taken because fecal leukocytes could be mistaken for amoebae. So that's it, and thank you so much for listening. And now I will give the floor to Miss Dorothy Planas Ablan for her report.
Hello everyone, and I am Dorothy P. Ablan. Now I will be the one to continue the last remaining part of this lesson, which is the pitfalls in the diagnosis of parasitic infections and the quality assurance in parasite microscopy. So, pitfalls in the diagnosis of parasitic infections. The laboratory diagnosis of parasitic infections plays an important role in the early detection of disease. In the Philippines, the diagnosis of parasitic infection is still much dependent on the ova and parasite, or what we call O and P examinations. In some instances, artifacts such as fungal spore, mite egg, plant cell, and pollen grain are mistaken as a parasite ova. Just like the example here, the first picture shows the fungal spore in a wet mouth stool that may look like a cyst of Entamoeba species. Second picture shows a mite egg in a formalin concentrated stool specimen that may look like a hookworm egg. So other artifacts like plant hair can be confused as helminth larvae. As you can see in the third picture, a plant cell in a concentrated wet mouth of stool may look like a helminth egg. So in the fourth picture, it is a pollen grain in a concentrated wet mouth of stool that may look like a fertilized egg of Ascaris lumbricoides. So the next slide is the first picture. a plant hair in a concentrated wet mouth of stool that may look like a hookworm or Strongyloides stercoralis larva. The second is the Howell jolly bodies and nucleated red blood cells, are sometimes misidentified as malaria parasite in a thin blood smear stain with GEMSA. And the third picture is a nucleated red blood cells that may look like a schizont of Plasmodium species. Lastly, a fungal spores of Helicosporium may also be mistaken as a microfilariae in a wet mouth stool that may look like a cyst of Entamoeba species. Now, these practices lead to inaccurate diagnosis of parasitic infection or false positives and inappropriate treatment of the patients. And now, let's move on to the quality assurance in a parasite microscopy. So for many parasitic conditions, as in helminth infection and malaria, microscopic examination is still considered to be the gold standard procedure. Note that the microscopies, however, differ in terms of basic training and skills. A physician, health manager, or scientific investigator should be assured the quality of the microscopic examination. So, in some occasions, the requesting party may also be interested to know about the burden infection of the patient. These can be determined by counting of the number of parasite or parasite ova in predetermined amount of sample, and reporting the intensity as number of parasite or ova per volume or weight of sample. In this practice, quantification of malaria parasite in the blood is more commonly requested. So the result is reported as the number of malaria parasites per microliter or of blood. So quantifying the parasite also helps these assessment of the efficacy of interventions such as chemotherapy in this control of specific parasite infection. The reliability of the results depends on the ability of the microscopies to identify and count stages of the parasites. So these can be assured by cross-checking of the same samples, whereby the reading of initial microscopies is compared to the reading of a reference microscopies. The reference microscopies must be blinded or must be no prior knowledge of the reading of the initial microscopies. For the malaria microscopy, the quality control may be done in the laboratory through blinded cross-checking of a minimum of 10 randomly selected slides. The first 5 reported as low density and the other 5 will be reported as negative. And this done by the trained validator or cross-checker at the end of each month. When the number of tests performed in 1 month is less than minimum sample size, all slides must be cross-checked. And for the soil-transmitted helminth infection and schistosomiasis, all negative slides and 10% of positive slides may be reread by a reference microscopies. The main drawback of this procedure is the technique may be laborious and not be feasible in many settings. So such as in a large scale of examination in surveillance and monitoring of the impact of the control programs. So it has been difficult to maintain the good quality for malaria microscopy, especially in peripheral laboratories. So for the current challenges in malaria microscopy includes lack of political commitment to support the development and expansion of the laboratory services. Second is the poor quality of microscopy, particularly at the peripheral level. Next is difficulties in maintaining microscopy facilities in a good order. Next is the logistic problems and high cost of maintaining adequate supplies and equipment. Next is the lack of adequate training and retraining of the laboratory staffs, the delay in providing results to clinical staffs, the lack of quality assurance and supervision of laboratory services, and lastly, inability to cope up with the workload of traditional system for the cross-checking of routinely taken malaria slides. This limitation can only be overcome by new health policies that acknowledge the importance of strengthening laboratory services, the need of adequate funding, and the implementation of a QA system such as adequate of staffs and resources, regular training and supervision of staffs and quality control of their task, accurate and timely slide collection, staining and reading, linked to the clinical diagnosis. Next is reliable results quickly provided to the clinicians. And lastly, provision of the logistic support for quality supply and equipment. So, this QA program designed by the World Health Organization that is used to assist the managers of National Malaria Control Program and the laboratory services to develop and maintain a sustainable malaria microscopy. So this program outlines structure based on a retraining, validation, and the development of the competency standard designed to ensure the quality of diagnosis necessarily for a successful malaria program. And when the validation of result is not feasible through the slide cross-checking, a certification scheme for microscopies may be considered, and microscopies will initially undergo the enhance their basic knowledge on the parasites. An intensive training on microscopic diagnosis of parasitic infections will be provide basic competency, which will be assessed through the theoretical and practical examination, that latter using prepared control slides. Sensitivity and specificity scores of the microscopies will be determined, and a certificate will be then awarded to the microscopists who passed the theoretical and practical examination administered by a panel of experts. And after certification, possible question and the veracity of results issued by microscopies may be minimized, and the the certified microscopies will also develop a higher level of confidence in performance of their laboratory work. So here is the reference of this discussion from the book of the Lesario. And that's the end of this video discussion. I hope you learned something from us. God bless you and thank you for listening.